Good evening and welcome to Intermountain Grand Rounds. I'm Paul Krakovitz, your host for today's Grand Rounds and the Chief Medical Officer for Intermountain Specialty-Based Care. Being a value-focused healthcare organization, nothing has challenged us more around quality and cost than the pandemic. And as we look to the future, the light that seems brightest right now for all of us at the end of the tunnel is the vaccine. As well as we're getting through the vaccine, it's important for us to have a good understanding of where we are with therapeutics. So we're very fortunate tonight to have Dr. Andy Pavia and Dr. Brandon Webb give us updates on therapeutic options as well as the vaccine. We will have some time at the end to answer questions, and at the end we will also give the CME credit code. By way of introduction, Dr. Pavia is a professor and practices mostly at Primary Children's. He is an infectious disease expert and is nationally and internationally known for many things around infectious disease, but most notably around pandemic response as well as vaccines. He's been a member of the National Vaccine Advisory Committee, the chair of the National Vaccine Advisory Committee Vaccine Safety Study Group. He has held multiple roles for our government, including chairing the 2009 Influenza Pandemic Subcommittee, as well as the sorted CDC and ACIP working groups. Dr. Brandon Webb is an active infectious disease physician who works primarily at Intermountain Medical Center. Dr. Webb is an associate professor of the Division of Infectious Disease and also the chair of our COVID-19 Therapeutics Committee. With this, I'd like to hand it over to Dr. Webb.
Thank you, Paul. I appreciate it and appreciate the opportunity to be here. I have no disclosures other than the fact that I am a lifelong Andy Pavia fan and happy to be the opening act for what I'm sure everyone's waiting for is the vaccines here. So just as a quick reminder, if you have questions as we go along, you're welcome to email sani.cairo@imail.org and she'll help make sure those surface for us to address. My couple of minutes here, I'd like to spend focusing really on a review of where we've come in the last few months with respect to therapeutics for COVID-19. And almost jokingly, I've described the pandemic as the period of time in medical history where I think we've found out more in as short a period of time about what doesn't work to treat a disease than ever before in medical history. And I say that somewhat jokingly, but partially in seriousness, because we knew right from the first days of the pandemic that respiratory viruses are very difficult to treat. I'm going to touch on a couple of different therapeutics in succession. We'll start with vitamins first, because this has been a very hot topic in the ambulatory setting. Most of you are probably aware that there's been a lot of interest around vitamin C since the days of Linus Pauling, and that's partly because of several characteristics, including the fact that we know vitamin C enhances interferon response, it decreases some cytokine production, and improves oxidative stress. And we have meta-analysis data that suggests that if you take vitamin C chronically over time, preemptively, you don't necessarily have fewer upper respiratory infections, but when you do, the symptom duration may be shorter. And that is really, to date, the sum total of our understanding of vitamin C and its potential in coronavirus, because we've studied it in other settings with the seasonal coronaviruses. Now, there is data in ARDS for a possible mortality benefit, but at this time, although there are more than 10 trials underway, there's currently no evidence supporting initiating vitamin C therapy in COVID-19. Vitamin D is another interesting association that has emerged we now, or we've long recognized that vitamin D contributes to immune function. And we now know that in multiple retrospective studies, hypovitaminosis D, or deficiency, has been associated with higher rates of infection and higher severity in deficiency. What we don't know yet is whether initiating vitamin D at the time of COVID-19 diagnosis improves outcomes at all. And in fact, there is one study that suggests that that's not the case. Zinc, finally, is another vitamin that has enjoyed a lot of interest in terms of respiratory infections. And there is meta-analysis data that suggests that zinc may shorten the duration of upper respiratory infection. However, supplementation over long periods of time with zinc is associated with copper deficiency, which can have some serious consequences. And here too, in COVID-19, we have no supporting evidence that it would improve outcomes. And it's worth noting that the NIH guidelines do recommend against supplementation over time for the prevention of COVID-19, again, citing this concern for copper deficiency. We'll move on to the next topic here. Remdesivir is an antiviral drug that was originally derived and developed for the treatment of Ebola, but has broad anti-RNA replication activity. It was studied in an inpatient setting in the ACT-1 trial, which was a placebo-controlled, randomized controlled trial. And in that trial, it showed a significant benefit in terms of improvement in time to recovery, in terms of improvement of symptoms. And in the subgroup of patients who were on supplemental oxygen but not critically ill, it did appear that there was a signal for mortality benefit. Now, a second study was recently published, the WHO Solidarity Trial, and I'll point out that in contrast, this was not a placebo-controlled trial. This was a pragmatic design comparing the remdesivir arm to a usual care cohort. And unlike in the ACT-1 trial, more than 50 or roughly 50% of the patients in the Solidarity Trial were on corticosteroids, and the spectrum of severity did tend to be a little bit higher in the Solidarity Trial. And here, we didn't see a mortality benefit. The FDA has approved remdesivir for use in hospitalized patients, but it's not currently approved for use in ambulatory patients. And we continue to use it in the hospital setting because we think that in patients who present to the hospital and may still be partially in the virologic phase, there is likely to be some role for antiviral therapy. And remdesivir in the setting of wards patients in particular may actually shorten the course. Now, there's still more to come. We need to understand this better, especially as it pertains to the treatment with corticosteroids. Now, I will highlight that in patients who are not in the hospital, remdesivir is not approved, but there is an outpatient trial looking at 3 days of remdesivir for outpatients in a high-risk population to see if early treatment may shorten the course. I'll also note that remdesivir is available in some settings in alternate care settings for patients who meet inpatient criteria, but during surge and contingency planning are cared for in settings other than the brick and mortar hospital. Corticosteroids have been one of the feel-good stories of the pandemic. We now have robust evidence that, especially when used in the critically ill, corticosteroids have a mortality benefit. Interestingly, there are now 4 large studies in less severe hospitalized patients that suggest that there is no benefit and potentially even a signal for harm when corticosteroids are given to patients who either are not hypoxic or don't have evidence of hyperinflammation. And for that reason, we recommend using corticosteroids in the critically ill and in a carefully selected non-critically ill hospital population who are either on supplemental oxygen or have evidence of the hyperinflammatory syndrome that complicates COVID-19. Now, I will also note that because steroids are well known to block the interferon response, which is critical for controlling the virus during the early phases, that there is potential that one of the reasons we may have seen that harm signal in less severe patients is that indeed perhaps those steroids are interfering with the normal immune response. And for that reason, we recommend against treating patients in the ambulatory setting with corticosteroids unless they have another indication. There's been a tremendous amount of interest around immunomodulators. And of the immunomodulators, perhaps none has enjoyed as much attention as tocilizumab, an IL-6 receptor antagonist. There are, per my count yesterday, 14 studies that have been published looking at tocilizumab, and the results have been mixed. The randomized controlled trials have generally showed little benefit, although the Krakovitz study, which we expect to see forthcoming in publication, suggested that there was indeed perhaps a shorter length of stay and length of ICU days. And a trial that has been hinted at but not published yet, the REMAP-CAP pragmatic study, did suggest that there was a mortality benefit. In contrast, there are about 6 retrospective studies in very highly selected populations with pronounced hyperinflammatory phenotype that do show benefit both in preventing respiratory failure, mechanical ventilation, and mortality. So at this point, a sum total appraisal of the literature suggests that if there is a role, it's very likely as adjunct therapy in steroid refractory patients who, despite several days of corticosteroids, are still advancing in their respiratory failure, and in whom there's evidence of the COVID hyperinflammatory syndrome. So baricitinib is another tyrosine kinase inhibitor that was studied in the ACT-2 trial in combination with remdesivir versus remdesivir alone. And in this study, there was in the treatment arm a signal for enhanced recovery, although it was modest, and this was most prominent in the patients who are on high-flow oxygen. There was a numerical mortality benefit observed, but this was not statistically significant. And when looked at in subgroup, patients who were already being treated with corticosteroids did not enjoy any additional benefit if baricitinib was added, which leads us to wonder then where the role of baricitinib may be, because it seems to have a very similar niche as corticosteroids, but is likely to be more expensive and less widely available. So at this point, it's not certain exactly where baricitinib will fit in our treatment algorithms. Now, a couple of notes about repurposed drugs. Hydroxychloroquine has had a lot of attention, and early on there was hope that because it blocked cell entry by changing the pH of the lysosome, that it may actually have a role early on as the virus hijacks our own respiratory cells. We also know, though, that it's an anti-inflammatory medication that does blunt the interferon response, similar to corticosteroids. And we now have robust evidence that hydroxychloroquine definitively does not benefit patients who are hospitalized. We also have several well-designed and carefully carried out outpatient and prophylactic studies that also failed to show benefit. And about 10 other studies are being currently meta-analyzed, and that work is in progress to see if a robust sample from a number of international centers reproduces the same lack of benefit that's been observed in the Spanish study and the Minnesota study. At this time, hydroxychloroquine is not recommended for treatment, neither in the hospital, nor in the pre-hospital setting, nor for prophylaxis. And then finally, I'll touch on ivermectin. Ivermectin is a very old and very well understood drug used for a number of parasitic infections. It does have well-known modest antiviral effect. The mechanism is thought to be blocking what we call docking mechanism. There's been a lot of attention recently on ivermectin, largely because of some very vocal and I think well-meaning proponents of this medication. But I think it's worth pointing out that as it stands right now, there is only very low quality evidence around the use of ivermectin in COVID-19. And in that body of low quality evidence, there are data on both sides, both suggesting that there is potentially no benefit at all if used early, and also some data that suggests that there may be some anti-inflammatory effect and benefit in hospitalized patients. But that was in a cohort prior to the advent of corticosteroids. There is a trial currently underway in Argentina in early disease that we'll be very interested in seeing when it matures. But at this point, ivermectin is not recommended neither for prophylaxis or for treatment outside of clinical trials. And it's also possible that any antiviral effect that ivermectin has is not seen in the doses typically used for parasitic disease. And finally, I'll finish up with a few words about monoclonal antibody therapy. This has been a therapy that we've looked forward to from the early days of the pandemic. Monoclonal antibodies mimic the natural humoral immunity, and in a setting where very few in the population possess humoral immunity, monoclonal antibodies hold promise for being an excellent therapy for neutralizing the virus. And we expect that this would also be most effective as early as possible in the symptom course. There are a number of these products currently in development. The first two to make it to market were a product called bamlanivimab, developed by Eli Lilly, and a cocktail medication, casirivimab imdevimab, which has been developed by Regeneron. And for those of you who are seeking CME credit, in order to get that tonight, you have to be able to pronounce both of these. Bamlanivimab has been studied in the inpatient setting, as has Regeneron. The bamlanivimab trial was in the ACTIV-2, Operation Warp Speed ACTIV-2 portfolio, ACTIV-3 portfolio. And in hospitalized patients, bamlanivimab actually did not show any clinical benefit. And that drug has now been discontinued as part of the ACTIV-3 platform. The Regeneron anti-spike trial is ongoing. However, in ambulatory patient populations, the BLAZE-1 and ambulatory spike trials for both of these products did both suggest that there was a decrease in viral burden, as well as an improvement in time to symptom recovery. But where the most interest falls is in the fact that in both of these studies, while underpowered, there was an observation that there was a decrease in medically attended visits, so emergency department visits or admissions to the hospital. And that effect was actually most pronounced in the higher risk strata. For example, in the Lilly study, in patients who are 65 or over or severely obese, the number of patients, the percentage who were admitted to the hospital were 15% in the placebo group and 4% in the treatment group. It's that observation that has motivated the FDA to issue emergency use authorization for both of these products. And they are now both authorized for use in the pre-hospital setting. They're both infusions, and both carry a nominal risk of infusion reaction, somewhere on the order of 2 to 5%, most of these being relatively mild. And there have, in both trials, been more severe infusion reactions rarely observed. For that reason, they're only authorized for administration in infusion centers, urgent cares, emergency departments, and some skilled nursing facilities where the capacity and the protocol to care for infusion reaction is in place. The issue with these is that currently the logistics around delivering an infusion medication that requires some training around how to do it and doing it safely with actively contagious early symptomatic COVID patients in intermingling with our normal clientele of infusion centers, which are often immunocompromised hosts, has proven more than challenging. And as it stands right now, although the drug is limited, our capacity to deliver the drug is even more limited. We know that these drugs are likely more effective in the highest risk strata. And for that reason, and also to match limited capacity and supply to the patients most likely to benefit, in Utah and in other states, eligibility criteria have been designed around risk stratification models that equitably match access to the drug with the patients who are at the greatest risk and therefore most likely to benefit. Here in Utah, our eligibility criteria is based on a risk calculator for hospitalization that was adapted from a model developed by the Cleveland Clinic and later validated in more than 22,000 Utahns, and did demonstrate that it is fairly accurate at risk stratifying patients. And that model is currently available on the Utah Coronavirus website on the landing page Novel Therapeutics, where both providers and patients can get more information about the eligibility criteria and the availability of the drug at various centers. And I'll finish up with showing this slide, which highlights the current state of Utah eligibility criteria. You'll note, and I'll let Dr. Pavia comment on this if he'd like to, that the eligibility for pediatrics is actually very, very restrictive because pediatric patients were not included in the randomized trials. And with that, I appreciate the opportunity and we'll be happy to take any questions offline. But for now, we'll get to the meat of the discussion and pass the ball to Andy Pavia for vaccines.
All right, well, thank you very much, Brandon. And I'm also a fan of Brandon Webb, so the feeling is mutual. Hopefully you can see my slides at this point. So what I'm going to do is give you a quick update on where we stand with vaccines in general, but really focus on the vaccine that we have at hand that we're going to be using very shortly, assuming my slides will advance. Let's see. Slight technical problem here. Let's see if we can solve it. There we go. These are my disclosures. I've received consulting fees over influenza-related issues from Glaxo and Squarius. So I want people to be aware that today is both a great day in science and in the battle against COVID, and a very somber day. We passed 300,000 deaths in the United States, almost 1.7 million deaths nationally, and at the same time, about 3 million doses of a vaccine that's highly effective against COVID-19 have been delivered to hospitals around the United States, almost exactly 12 months after the first reports of serious disease emerged in Wuhan, China. And so what I'm going to do is briefly go over the vaccine landscape because it's important to know about what other vaccines are coming. I'm going to spend a few minutes just talking about how mRNA vaccines work and why we shouldn't be scared of them. And then I'm going to dive deeply into the Pfizer vaccine efficacy and safety so that people can inform their patients and make informed decisions for themselves. So versions of this slide keep showing up all the time, but this is updated as of today. The Moderna and the BioNTech Pfizer vaccines are mRNA vaccines that have completed their phase 3 trials. As of Saturday, the Pfizer vaccine has an FDA emergency use authorization, and Thursday, the FDA will be meeting to consider giving a similar authorization to the Moderna vaccine, and we expect that it's going to be granted. And we really want to know what's happening next because these two vaccines aren't going to produce enough vaccine to end the epidemic in the United States by themselves. So the Oxford AstraZeneca vaccine, which is a chimp adenovirus vaccine, was thought to be very promising, but it's had a bit of a setback because of some complex data that they announced from their phase 2 and phase 3 trials. They accidentally did a low dose, high dose arm, which they did not intend to do, of about 3,000 patients, and their standard dose in some 25,000 patients. The standard only showed 62% efficacy, but in that small number of patients who were primed with a smaller dose and got the higher dose, they had 90% efficacy. This may be a fluke, and it may be real, but it's meant that the FDA has asked Oxford to go back to the drawing board to redo trials to really solidify that. Probably the next vaccine we're going to see that we're excited about is the Johnson & Johnson vaccine. It's an Ad26 vaccine that only needs a single dose, so that's a huge advantage. It's fully enrolled as of Friday last, and we expect to see results out in January, and if they're good, that should move to fairly rapid licensure. The Novavax vaccine is a protein nanoparticle with an adjuvant. It's enrolling in phase 3 now. It should potentially have full enrollment in the middle of the winter, so that may follow soon after. The Sinovac vaccine is being used in several countries, despite the fact there really are no published phase 3 trials to give us a complete picture of its activity. Following up behind it are some candidates that look pretty good. Merck has 2 vaccine candidates in production. There's a measles virus and a VSV vectored vaccine. They're only in phase 1, but the immunogenicity looks quite good. Sanofi GSK are partnering on a vaccine that we had high hopes for that's had a bit of a setback because they announced last Thursday that they had lower than expected immunogenicity in the older cohort. So they're going to go back and look at different dosing for older patients, and that's going to delay that one. There's a DNA vaccine and then a Vaxart adenovirus vaccine that are also in human trials in the United States that we may see in the summer. We're going to need all of these vaccines because we're going to need something in the range of 650 million doses of vaccine in the United States, and the first 2 vaccines won't provide that. But the first 2 vaccines are mRNA vaccines, and some of you have heard me talk about this before. But of course, mRNA is the template on which cells form proteins. And if you just give mRNA alone, it ends up being a very strong stimulant to the innate immune system. It doesn't enter cells. It causes a lot of inflammatory response. So there have been some brilliant modifications, particularly pioneered by the team at BioNTech, the husband and wife team that did this, that have made it into an effective vaccine. They've modified the protein in the middle by doing codon optimization of the spike protein to keep it stuck in the pre-fusion conformation, which is the conformation that we want for immunogenicity. And then they modify the mRNA molecule in a number of other ways, including this poly-A tail and a cap that reduce the immunogenicity in terms of an unwanted innate immune response, but allow it to create an appropriate immune response. It's formulated in lipid nanoparticles, which leads the follicular dendritic cells, shown here as this sort of spidery thing, to take them up preferentially. Within the cell, the spike protein is made, it's processed, expressed on the surface in a number of different MHC classifications. But what's important is that leads to both a CD4 and a CD8 response. It leads to both an antibody and a cell-mediated immune response with what appears to be long-lived memory. So the Moderna vaccine is a good example of an mRNA vaccine. It's also lipid nanoparticle encapsulated. It also requires 2 doses. This one is 28 days apart. The big advantage that the Moderna vaccine has is that it can be stored for long periods of time in ordinary freezer temperatures and is relatively stable for a while in the refrigerator. It's fully enrolled its trial, and on November 30th, they released results by press release. You've heard me editorialize about science by press release, but their press release was actually pretty comprehensive, and it reported on 185 cases in controls, 11 among the vaccinees, giving a point estimate of 94.1% efficacy, almost identical to what you're going to see for the Pfizer data I'm about to show you. More importantly, they had no cases of severe disease among the vaccinees and 11 among the control group. And they did a good job of enrolling older patients. 23% were over 65, and 37% of the enrollees were from community of color. So next Thursday, VRPAC will review all the data and we'll learn more about it. What I'm going to share with you about the Pfizer vaccine in detail comes largely from the VRPAC presentations. And VRPAC is the Vaccine and Related Biologicals Advisory Committee. It is an independent group of highly expert vaccinologists and vaccine scientists that reviews the data. Pfizer submitted their data about 2 weeks earlier, and the FDA reanalyzed everything. So what I'm going to show you comes largely from two documents. One is the FDA briefing document, which is their own analysis of the data. It's about 53 pages. And the Pfizer data package, which is about 105 pages. You can download these from the FDA website and read them all yourself. If you don't want to spend all night doing that, you can also read an article in the New England Journal that was published last Thursday. It's sort of the Cliff Notes version. And when you pore over the longer documents, you'll see how much gets left out of a journal article. The Pfizer vaccine, I think everyone knows by now, is a codon-optimized mRNA. It's 2 doses 3 weeks apart. It now has an emergency use authorization issued Friday night. And that emergency use authorization is to prevent SARS-CoV-2 infections in persons 16 years of age and older. The logistics, as everyone has heard, are kind of daunting. To transport it at -70, it requires being shipped in these specially designed containers. Each layer of the container contains 975 doses, and there's 60 pounds of dry ice in these containers. The vaccine is, when it's thawed, is only stable for 120 hours. So you have to use all 1,000 doses fairly rapidly. And this creates a lot of logistic challenges, which is why this vaccine is going to be used largely at major centers that can handle it and that have the ability to either reload the dry ice or have a -80 freezer. In the phase 1/2 studies, these vaccines showed really good immunogenicity. In the upper left, you see the neutralizing antibody levels, and they exceed those seen in patients recovering from symptomatic disease, which is the target we really wanted to go for. But more importantly, in the lower right, you see a Th1 bias CD4 and CD8 response, and that response is quite a bit higher than although the numbers are small, than what was seen in the T cell response among the recovered patients. So that was the preclinical data. Now we're going to move into the clinical data from the phase 2/3 study. Just to orient you to the large study, the 459-1001 study, it was an adaptive trial that began as a phase 2 study. So the first 360 patients were intensively studied. They were restricted to people over 18 years of age. And when that showed good immunogenicity, the study was expanded first to include a much larger portion for efficacy. And then they included 16 to 17-year-olds. And finally, they've just begun in late November to include 12 to 15-year-olds. There's a little bit of data on the 16 to 17-year-olds, but nothing yet to report on the younger age group. The data were cut off on November 14th, and that'll be important in a couple of minutes. There are, in all of these trials, different populations that are used for the different analyses. I won't bore you with too many details about this, except to say that the efficacy population are those that were followed for at least 2 months, and that's about 40,000. The safety population had to include those who had at least 2 months of follow-up from their second dose, and that turns out to be about 37,000. But some of the analyses focused on everyone who was enrolled in the trial, regardless of follow-up, and that gives you an intent to treat population as well. There are different endpoints for this trial. The primary endpoint that Pfizer picked was a little bit odd. It was the incidence of COVID-19 in vaccine recipients and placebo that excluded those with any evidence of prior infection and who had a case diagnosed within 7 days. There was also an analysis done using the more standard FDA NIH definition of cases that occurred after 14 days, and another analysis that included those who had evidence of past infection. And these all really gave the same results, so I'm not going to focus on them, but I will show you data on prevention of severe disease, because that's really what we want to prevent, and that's an important endpoint. The demographic slides are important in any of these studies because you want to know whether the population studied is the same population you want to treat. And in this case, they did a pretty good job. The vaccinees included about 9% African American or Black, and about 28% of the patients were Hispanic or Latino. Notice that some of the ethnic groups that are represented in Utah were not well represented overall in the trial. The age distribution is shown below, and you can see that there's only 153 people 16 and 17 years old who are included, and only about 1,800 people over 75. So more to learn in those age groups. About 35% of patients had a BMI of greater than 30%, and that was not because of specific recruiting. It's just unfortunately a reflection of people at risk in the United States. All right, now for the efficacy. And this, quite honestly, when we first saw it, blew us out of the water. There are very few vaccines that have efficacy of 95%. Measles with 2 doses exceeds that. Shingrix with 2 doses is about at 95%. Nothing else really gets to that level. One of the ways to really understand the efficacy is through this kind of slide. This shows the cumulative incidence of symptomatic infection from the time of the first vaccine. And you can see that for the first 14 days, the curves overlap perfectly. And then they begin to separate 14 days after the first dose. This suggests that there's at least some immunologic protection after about 14 days after the first dose, which is about exactly when you'd expect antibodies levels to start to rise. And then after 21 days, you can see very low accumulation of new cases among the vaccinees. So does it work to give a single dose? The point estimate of efficacy is about 50% for that first month. But remember that everyone in this trial went on to get a second dose. So it really cannot tell us anything about the efficacy of single dose. And even though people have talked about whether that would allow us to vaccinate more people, we just don't know whether it would work. Here is the meat of the presentation. You can doze off after this. Primary efficacy analysis included about 170 cases, relatively small number. There will be more cases accumulating in the follow-up, but the estimate of the efficacy was 95% with a lower 95% confidence interval of 90%. So this vaccine is at least 90% efficacious, even if you do an enormous trial. The efficacy was similar in those younger than 55 and those over than 55. And then in this slide, if you look in the box, you can see that, again, the efficacy was virtually identical throughout men and women, people from different racial groups, ethnic groups, and in the different areas where the trials were undertaken. And here you see the efficacy among people without any comorbidity in the top line, 94%, 94.7%, and then people with varying comorbidities. And strikingly, the efficacy is good in all of these groups. Now, people who were undergoing active cancer treatment who were severely immunosuppressed were not included, but among these participants with underlying diseases, the efficacy remained in the 95% range. The severe disease analysis is a little bit more complicated because there are 2 definitions which give you 2 rather different numbers, but I think the overall sense is about the same. The FDA definition of severe disease gives you a total of 1 case among vaccinees and 9 among placebo recipients, but many of these severe cases occurred before the second dose. So if you look at just those who had disease after receiving 2 doses, there was 1 case in the recipients and 3 in the placebo recipients. Both of these give you an estimate of vaccine efficacy that's significant, well, that is noticeable, but it is not statistically very precise. And for the standard definition, it is not statistically significant. The FDA uses a CDC definition, which is, CDC has a definition which the FDA does not use, which was also used in the analysis. And this gives you 1 case in the vaccine recipients and 14 in the placebo recipients, which does give you a statistically significant efficacy estimate for people getting severe protection against severe disease of 92%. So however we slice it, there's at least a strong signal of protection from severe disease. What we don't have, and somebody will ask about this no doubt, is any data as of right now about protection against asymptomatic shedding. And that's that is data that we're going to hopefully get answers on in the January-February time point. So what about people who were previously infected with the virus who got vaccinated anyway? If you knew that you'd been infected, you were not supposed to sign up for the trial, but when they tested people for antibody and for PCR, they did actually find that about 1,300 people were either actively or previously infected. There were no increased side effects or safety signals in that group, but what they did see is that among both placebo recipients and people who got the vaccine, there were 9 cases and 10 cases respectively among people who'd been previously infected. This doesn't tell us much about efficacy, but what it does confirm is that people who've been previously infected can be reinfected. This is something we know, but this gives us an actual rate of about 1 per patient year, 1 per 100 patient year, or 0.1 per patient year. All right, since we're all lining up to get the vaccine, hopefully enthusiastically, you want to know what the side effects are going to look like and what the safety signal is going to look like. And I should begin probably by saying that I've pored over this data a lot, and I can't wait to get my vaccine. So it's important to realize that there's only 5 months of follow-up in the group with the longest follow-up from July 27th to November 14th. But most people have about 2 months of follow-up. There will be more data coming out with 6-month follow-up and 2-year follow-up, and there's a number of other surveillance systems I'll touch on at the end. Overall, about 82% of people complained for either the first or second dose of some local reaction. After the first dose, it was about 50% had mild pain, and about 30% ranked their pain as moderate. Redness and swelling were less. I did look up the data for Shingrix for comparison if you've given or had that vaccine, and the local reactions are substantially higher for Shingrix, so it gives you something to use as a benchmark. Overall, about 72% of people at some point reported some systemic side effect from the vaccine, but let me show you more detail here. Just to orient you, the top are the vaccine recipients, the bottom are the placebo recipients. In each bar, there are 2 bars, one for the younger folks who are under 55 and one for those 55 and older. And you can see that after dose 1, about half of the people reported fatigue in the younger age group, 34% in the older age group, and fever was seen in under 4% in the young ones and 1.4% in the silver fox and mature crowd. It's important to note that the placebo recipients had a lot of fatigue, a lot of headache, a lot of chills, and equal amounts of diarrhea. I think the fatigue just reflects 2020, and anybody who wasn't reporting fatigue was probably not filling out the form correctly. This is important though. This is the second dose, and side effects go up with the second dose. Now, fever is reported in 16% of the younger group and 11% of the older group. I'll show you more details in a second. Fatigue in 60% and 50% respectively. And there's more headache. There is more myalgia and arthralgia. Most in green are mild or moderate. You'll see a little bit of the gray stripe, which is severe, but remember that severe is defined as being bad enough to cause you to change your activity. So it doesn't necessarily mean it was very severe, but it meant that people took a day off from exercise or from work or avoided other activities. The side effects mostly occur in the first 24 hours or so after vaccine, usually the day after vaccination, as shown here on day 2. They tend to last 24 to 48 hours. It's important to anticipate that people will have some side effects. Tylenol can be used to blunt the side effects, but prophylactic use is not recommended. And here in more detail are the fever data, because I think this is helpful in thinking about how we're going to distinguish somebody who we think might have COVID-19 or another illness versus a vaccine side effect, you can see that although 16% of the young people had a fever of greater than 38, that about 2/3 of them had a fever that was only 38.0 to 38.4, so quite low grade, and about 6% had higher fevers. This is after dose 2, the more reactogenic dose. And for people in the older age group, only 3% had fevers that exceeded 38.4. And there's the fatigue numbers again. So that's side effects. Now we want to talk about, what are potentially serious safety signals, and there really were none so far. Serious adverse events include anything that results in hospitalization or death. There were 8 in the vaccine recipients. Actually, most of those were appendicitis, and 4 in the placebo recipients. There were 2 deaths in the vaccine group and 4 in the placebo recipients, all thought to be not associated. The one thing that did crop up was, as a statistically significant spontaneous reported issue, was lymphadenopathy in 64 patients. That makes sense and was felt to be totally plausible. There were 4 cases of Bell's palsy in the vaccine recipients, none in the placebo group, but 4 cases among 22,000 people was felt to be within the background rate. So at this point, we can't make any conclusions about whether there's a causal relationship or not. Women who were pregnant were excluded from the study, so we have very limited data on pregnancy. There were 23 pregnancies diagnosed after the receipt of vaccine, and there were no serious pregnancy outcomes in the vaccine recipients, and in the placebo group, there was 1 spontaneous abortion and 1 retained products of conception. So we really can't say much about the safety, and I will come back to what we can tell pregnant women in the next slide. But the one thing that cropped up immediately after the vaccine was used in Great Britain was this case report of 2 people who developed anaphylactic or anaphylactoid reactions. They were healthcare workers. They both had a history of severe anaphylaxis. They carried epinephrine injectors with them. And we don't know what they were responding to at this point. There had been no severe cases of anaphylaxis during the trial. So how are we going to handle this? The FDA issued a warning that said, do not use for patients with a history of anaphylaxis to any of the vaccine ingredients. And those are listed, you can look them up. ACIP went a little bit further and said anaphylaxis to any other vaccine or injectable product is a precaution, not a contraindication. And these people should be kept in the area where do the vaccination for 30 minutes and observed, and you should have the ability to start treatment for anaphylaxis at a vaccine site. So here's some other key recommendations that ACIP put out. And ACIP gives us sort of our practical, how do we use vaccines advice. So in terms of people with prior documented infection, they say that we should not be doing serologic testing for the purpose of making vaccine related decisions. And current evidence suggests that reinfection is uncommon in the 90 days after initial infection. Persons with a documented acute infection the preceding 90 days may choose to defer vaccination until the end of this period. People who have acute COVID-19 can be vaccinated once they're out of quarantine, so after 10 or 20 days, depending on severity. Pregnancy and breastfeeding are obviously of great concern since many of our care providers are pregnant or trying to get pregnant. And I think in a very enlightened way, the recommendation was much more progressive. In the past, we've often said, we don't have any data, so you can't have it as a pregnant person. But instead, the recommendation is that we provide the information that we really have no data on the safety of COVID-19 vaccines in pregnant women. But if a woman is a part of a group who is recommended to receive COVID-19 vaccine, she may choose to be vaccinated after weighing the risks of COVID-19, which has known risks that are increased in pregnancy, and the risks of the vaccine, which are unknown. What we can say is that no other vaccine has been demonstrated to be unsafe for pregnant women, with the possible exception of live vaccines, which we carefully avoid. But there is a theoretical concern about high fever in the first trimester. But we don't know what we don't know about long-term safety. 44,000 patients is not enough to find a 1 in a million side effect, of course. So there are really extensive systems that were developed in 2009 for swine flu that have been adapted to and expanded to give us a really good ability to pick up any safety signals that exist. We divide this into two systems. One are those that are looking for signal detection. So any report of an event that might possibly have a temporal association with a vaccine, and then what are called signal assessment studies, where controls can be used. So you can actually start to decide whether or not there is reason to believe that there's a causal relationship. And I won't go through all of these, except for one that you can see in pink on the near the center of the slide, called the VSafe system, because we're all going to be invited to participate. This is a text-based reporting system for early side effects as well as for possible serious adverse events that you can sign up for. There will be, you'll receive information about it. You can scan the QR code with your phone and sign up, and then you get a text-based request to report on your side effects for the first week, and then a check-in at different intervals to see if there's anything more severe. So with that, just a quick reminder of how this rollout is going to work nationally and in Utah. We're now in phase 1A, which is healthcare personnel and long-term care facility residents. And the numbers here are there are about 130,000 healthcare practitioners, healthcare personnel defined broadly in the state of Utah, and about 28,000 long-term care facility residents. We anticipate getting about 150 to 160,000 doses by the end of December. So theoretically, there should be enough vaccine to give the first dose to everyone in phase 1A. Phase 1B are essential workers, and that definition can be a little bit controversial, and that's being decided on a state-by-state basis. In Utah, the numbers are about 160,000 essential workers, 94,000 teachers, 14,000 first responders. And you can see that we're going to need more vaccine to get into this group. So that's looking like a January, February timeframe. And then phase 1C are those at higher risk because of medical conditions or because of age. That's about 650,000 people. So we're going to need more vaccine to get there. Lots of unanswered but critical questions. How long does immunity last? What are the correlates of protection? Does the vaccine protect you against getting asymptomatically infected and having ongoing transmission? We don't know the answer to any of these questions yet, but I don't think we're going to have to wait too terribly long. So I want to end there, and I'm not going to say anything about light at the end of the tunnel. That can always be an oncoming train, but use an analogy I prefer, which is, we can see the top of the mountain. So we know where we're going and that there's an end to this climb, but there's a lot more climbing to do. There's a lot more danger ahead on the ridge. And in the meantime, we really are going to have to double down on our public health measures and our community mitigation. So with that, I'm going to end, and hopefully we'll have some time for questions, but I'll leave this slide up, which will allow you to claim your CME credit. The credit code is 6493, and there are 3 ways you can ask for credit. You can use the Cloud CME app, you can visit the website, or you can text that code to 844-989-1332. That's 844-989-1332. Operators are standing by. And with that, I'm going to turn it back over to Paul for questions.
Andy and Brandon, Dr. Webb, Dr. Pavia, thank you so much. Incredible information that you guys gave in a way that even a pediatric otolaryngologist could understand. So thank you. There are a number of questions. The first question is, do antipyretics or analgesics taken around the time of injection have any interference with vaccine efficacy?
It's a great question. We don't know the answer for these vaccines. There is a little bit of data that is not completely accepted as scientific fact that pre-treatment with NSAIDs and possibly with antipyretics blunts the immune response to some childhood vaccines. That may or may not have any applicability to adults, but CDC is recommending that while people should be advised to use them to treat symptoms as they're starting to develop, you should not take them before the injection and not until you start to feel the potential of some side effect.
Thank you, Dr. Pavia. I think a big question for people in Intermountain is, how is this rollout going to work, and how are we going to actually start getting the vaccine placed into our arms? I will say, give a shout out to our physician and APP volunteers. There's been over 200 physicians and APPs in Intermountain that have volunteered to help give vaccine, which is incredible. It allows our MAs and nurses to be at the bedside where we need them to fight the actual disease. We do have Joe Franson with us, and he is a continuous improvement expert and has been deep into the implementation. Joe, if you are able to come off and mute, could you give us a quick update and kind of plan on where we're going with how we're giving the vaccine?
Sure, Paul. Yeah, it's just kind of a very quick overview on the Intermountain vaccine distribution plans. We actually had our first 2 sites receive vaccine today. LDS Hospital and Utah Valley Hospital both received their first shipments today. We expect another couple of sites, Intermountain Medical Center and St. George, to receive their initial allotments tomorrow. And we have a couple of other hospitals that will start coming on. We are in the process of finalizing enrollment with the Utah Department of Health for all of our hospitals to be vaccine providers for the CDC over the next couple of weeks to vaccinate healthcare workers, and then we will start to transition into planning for offering this to the community and to our patients, and that'll be more in the outpatient setting through our outpatient pharmacies and our clinics. The initial vaccination sites will start up here over the next couple of days. LDS Hospital is going to start tomorrow, and then additional hospitals will start over the next couple of days with the intent of distributing our initial allotment of about 10,000 doses by the end of the year. So we'll stagger that with the holidays coming up over the next couple of weeks, but we'll be able to get that first dose to 10,000 by the end of the year, and then that starts the clock ticking on the 21-day time period to get a second dose into that group. Over that same time, we'll expect to have several other hospitals stood up and receiving vaccine. A lot of that's going to depend on timing with Moderna to get vaccine to sites that don't have the ultra-low freezers with the requirements that the Pfizer product has. So all of those sites will continue to spin up, and we'll start moving forward and working through our Intermountain caregivers and providers based on risk of exposure. So we've developed kind of an algorithm for determining which units and which provider specialties would stand up first, and then we would just work down that list, and we've been able to, at least at those initial sites, be able to cover a large majority of our highest risk employees and staff and providers, and we'll continue to work down that list. UDOH has indicated to us that they'll supply vaccine about as quickly as we can use it and as quickly as the supply chain from Pfizer will allow. So we anticipate starting here in the next few days and working as quickly as we can to start moving through this first phase.
Joe, thank you for that update. And of course, those that are non-Intermountain who are listening, please take the advice of your institutions. I know the U has a plan as well, and I'm sure there is for Stewart and HCA hospitals as well. Brandon, quick question for you around prophylaxis for COVID fungal concerns and using mycofungin prophylaxis. Could you address what the up-to-date thought process was around that?
Yeah, you bet. There are a number of centers around the world where fungal infections in general are more common than we see in the arid environment of the Mountain West. And so fortunately, across the board, we have less fungal infections than other places in the world. But in areas where they have higher rates of fungal infections, there has been a significant amount of what they have now termed COVID-associated aspergillosis. And we have seen some cases here in Utah, but low incidence. So at this point, we're recommending a screening process rather than a prophylactic process. And with with fungal biomarker screening, we can identify patients who may merit antifungal therapies. And we have a pretty good sense of who these patients are. They're patients who are on the ventilator, who've been on steroids or other immunomodulators, and who are still lymphopenic. And by having visibility of which patients they are and using screening with fungal biomarkers, I think we have a good process to identify which patients might benefit from antifungal therapy.
Thank you, Dr. Webb. Well, Dr. Pavia, Dr. Webb, and Joe Franson, again, thank you so much for this presentation. It was enlightening. This will be recorded and will be kept on YouTube. So for those people, you want to tell your friends who want to watch this asynchronously, it'll be available for everyone. So again, thank you, everyone, and have a good night.